The work we do here at Compassion Medical Group is a part of the overall process of drug development. This process lasts years and is extremely expensive to conduct. Many people with many differing skills and backgrounds are involved in drug development. Let me give you an example of a typical drug development process for a novel new drug involved, let us say, in the treatment of rheumatoid arthritis, which we will call RA. This process may vary slightly for different drugs and is a little different for medical devices, but the overall process is similar. The first step in drug development is the identification of a condition for which a new treatment is needed. Conditions may be the result of disease, congenital conditions, trauma or accident, or even a situation that might be considered a deficit, such as inordinately short stature or poor night vision. When a condition is identified that needs medical attention, researchers begin to look for potential therapies. In a case where a company becomes interested in a medical therapy, such as a new anti-inflammatory drug for RA, the laboratory researchers begin to look for new chemical compounds that might be promising as a therapy. New compounds often are small molecules isolated from natural products such as plants or bacteria. Recent advances in computer modeling now allow scientists to design custom chemical molecules for specific purposes. The laboratory process of chemical synthesis or isolation, modification of the molecule to ensure it is stable and absorbed into the body, and compounded into a pill or injectable form may take 1 or 2 years. Once a prospective therapeutic has been compounded, it must be tested in a non-human system for safety. This portion of the drug development process is called the preclinical trials. Most often, the compound will be given to animal models to ensure safety and to look for potential effectiveness. In the case we are discussing, we might use a strain of rats that have arthritic problems that seem similar to human RA. We will treat the rats with our new compound and look for endpoints such as improved mobility or decreased joint swelling. If the preclinical trials show that the drug is free from toxic effects and shows some promise in mitigating the effects of arthritis in the rats, we can submit a request to try our drug in humans. Preclinical testing may take from 1 to 2 years to complete. With acceptable preclinical data, we submit an investigational new drug application called an IND to the FDA. The FDA will examine the preclinical data and level of need versus the level of risk of our treatment and may approve our IND. The IND is an exemption for a company that allows them to do 2 things. Firstly, it allows the company to administer a non-approved drug to human beings. Secondly, it allows a company to ship unapproved drugs across state lines to clinical trial sites. Shipping an unapproved drug across state lines is a serious violation of the Interstate Commerce Act. The IND does not allow a company to sell a drug for a profit. The exemption is only allowed for research purposes. The FDA oversight is focused on 2 primary goals: that a new therapy be safe and effective. Once the company has an IND, it can begin Phase 1 trials. Phase 1 trials are very limited trials with 2 major purposes. The first is to assess the safety of the drug in human beings. Since human physiology can vary significantly from the model animal, safety has to be tested in humans. The second purpose of a Phase 1 trial is to get some estimate of the necessary dose of a drug to be used in further trials. As before, animal models may metabolize or eliminate drugs very differently than human beings, so appropriate doses of a drug in rodents may be very different for humans. Phase 1 trials often enroll healthy subjects, not people who might be compromised by disease, since the effects of the drug under study are largely unknown. Phase 1 trials usually have a small number of subjects, perhaps only 10 subjects. When the data is examined, the lack of any adverse side effects due to the administration of the drug is the most important factor examined by the FDA. Researchers look at the data for hints to useful doses of the drugs. The Phase 1 trial may take 1 to 2 years to complete. The FDA will examine the data and, if considered acceptable, will approve the IND for Phase 2. In Phase 2, the drug will have its first real scrutiny to assess whether it might become a useful therapy. Phase 2 trials are larger in scope, both scientifically and subject-wise. Phase 2 trials will often enroll up to 100 subjects. In this phase of the research, the safety of the drug will still be examined, but since the Phase 1 trial indicated that the drug appeared to be safe for use in people, Phase 2 focuses on the drug's effectiveness. The data must support the hypothesis that the drug will improve whatever outcomes the researchers have chosen as markers for effectiveness. If a therapy is not better than currently available therapies, there is no rationale to allow people to be exposed to as yet undetected potential dangers from long-term exposure. When the Phase 2 data is collected and reduced, the company should have compelling evidence that the new drug is likely to be safe and effective for its prescribed indication. The Phase 2 trials may take between 1 and 3 years to complete. Once finished, the Phase 2 data is submitted to the FDA for review. If the FDA is convinced by the Phase 2 data that the drug appears to be safe and effective in limited trials, the IND will be approved for Phase 3 trials. Phase 3 trials are large multi-center studies. Since the therapeutic dosage was determined for the drug in Phase 2, safety and effectiveness can be extended to a broader demographic. Because of statistical considerations and the logistics of locating and enrolling enough qualified subjects, Phase 3 trials typically employ many individual research sites that each enroll a small number of subjects. Often, 100 individual sites will each enroll 10 subjects for a total enrollment of 1,000 subjects. There are a number of ways that the trials can be performed, but for simplicity's sake, when discussing our RA drug, subjects will be randomized into 2 groups. One group will receive our investigational drug, and they will be called the treatment group or the experimental group. The other subjects will receive a standard drug for RA that is commonly prescribed. The data will allow us to determine if our new drug may be more effective for treating RA than the approved drug. In our case, subjects with RA are enrolled and given either the investigational or approved drug without either the subjects or the researchers knowing which drug they are receiving. This is called a double-blind design, and we will discuss it and other designs on a later date. After the data are collected, the data are tabulated, and statistical analysis will test the effectiveness and safety of the investigational drug. This data will be transmitted to the FDA with the request for approval of the drug for sale. The Phase 3 study may take from 2 to 4 years. The FDA will convene a panel of scientists and statisticians with expertise in the field that the drug represents to review the data. They will review the data for safety and effectiveness. The panel can recommend 3 possible dispositions for the application. 1, approval, 2, denial, or 3, requiring additional research. In the case of approval, The company is given permission to label the drug for the indication approved by the FDA. In the case of denial, the drug is considered to be unfit for human prescription and may not be marketed. In the third scenario, the company needs to gather more clinical data to answer some specific concern that the panel has as to safety or effectiveness. Companies can appeal decisions not in their favor to the Commissioner of the FDA or the Secretary of Health and Human Services. After a drug is approved for marketing, the FDA may, if it has cause, require a Phase 4 trial, usually called a post-marketing trial. This study will provide follow-up data over a longer period of time and in a real-world situation where there is less control over those receiving the drug. It allows the FDA to see the effect of other drugs or lifestyle issues on those taking the newly approved drug. The most current data on novel, new investigational drugs shows that the average amount of time required from discovery of a potential new drug at the laboratory bench until it is on the shelf at the pharmacy is between 10 and 13 years and costs the company approximately $900 million. Most drugs never make it to market because of safety or effectiveness issues. Some estimates say that for every 3,000 new drugs tested, only one will ever make it to the marketplace. The burden on the company is compounded by the fact that the patent on a drug, which starts counting down at the time of the drug's discovery, is 20 years. This means that companies have 7 to 10 years to recover their investments in approved drugs, cover losses in failed drugs, and make a profit to sustain the company. For this reason, the data collected by the clinical researcher in the drug trials are the linchpin of the drug discovery process, and the clinical researcher's outcomes have much to do with a company's sustained existence. You are entering a very important field. To help familiarize yourself with the steps in this long process, Dr. Blanson wants you to do some research. He feels it would be helpful for you to study some of the documentation required by the FDA during the approval procedure. You should get an email from him soon outlining details of what information he wants you to gather.